Okay, this is the lecture for microbiology. This is the first in a series of 2 lectures for the week of Monday, January 23rd. Where we left off, we're on Chapter 4, Morphology and Taxonomy. And here are a lot of different shapes of bacteria. And I really hate to say this, but you need to be able to identify and know what all of these shapes are. So this is a good slide for review. Here's coccus up top, bacillus, which is a rod, and then curved and spiral forms. Primarily with curved and spiral forms, we talk about spirilla, which are rigid spirals, and spirochetes down here, which are flexible spirals. Spirochetes have their flagella inside their periplasmic space, so they wriggle around. The flagella are invisible because they're inside the cell. Vibrios are curved rods, and let's see, different. You can have different variants of rods. There are stubby rods, we call those coccobacilli, pill-shaped rods, palisades arrangement. This is where 2 cells are hinged. Just in 2 rods, and so they actually make sort of a hinged arrangement. Sometimes you'll see some that form filaments, and then obviously, like we talked about before, some will form spores. In terms of coccus, if you have 2 together, that's a diplococcus. If 4 is a tetrad, 8 or greater in a sort of a close packed arrangement, like a cubic arrangement, is called sarcinae. You can have streptococci, which are in chain, and then you can have staphylococci, which are in clusters. Okay. So here are some things to remember. Bacillus is rod-shaped, coccus is spherical, vibrio is curved. Spirillum is spiral. A spirillum is a rigid spiral, and a spirochete is a spiral spring, or flexible, because the spirochete has a flagella inside of its body. Pleomorphism, there are cells in the single species that can change shape or size depending on the condition. Diplococci, 2 coccus cells in pairs. Tetrads are 4. Sarcinae are multiples of 2. Staphylococci, staph means cluster, so that would be a cluster of cocci. Streptococci, strep means chain, so this would be chains of cocci. Palisades arrangement, cells hinged on one side. Okay, and here's the diplococcus. Here's a bacillus, just a artist rendition. These are vibrio cells, curved rods, and then this is streptococci. Okay, now we have to name bacteria, so we're looking at the taxonomy of bacteria. This is based on genotype and phenotype. Genotype is from the ribosomal RNA small subunit sequence, basically the 16S sequence, and that's the basis for phylogeny. That will spill over into taxonomy as well, especially when you have very, very new species that are being characterized. Then routinely the 16S sequence is obtained. In terms of phenotypic traits, a lot of the bacteria that we view in the laboratory are named based on their phenotype, their bacterial shape, their smell, their cultural behavior, their biochemical reactions. And so what we see now, because we can do genotyping very easily, is that in modern taxonomy there are both aspects of naming organisms. Okay. In terms of prokaryotes, and this says kingdom prokaryota, scratch that out. Let's say in prokaryotes in general, prokaryota is no longer a kingdom. That has changed. Okay, so in prokaryotes, there are 3 kingdoms gracilocutes, which are gram-negative, firmicutes, which are gram-positive, tenoracutes that don't have a cell wall. Okay, so circle those first three. Those are all bacteria. They're in the domain bacteria. And then the last one is actually the domain archaea. These are mendocicutes. And this is the domain archaea. They have unusual cell walls and unusual nutritional habits. Gram-negative is important medically because of the lipopolysaccharide on the outer membrane, which causes fever and shock. Gram-positive bacteria because of their strong cell wall. Aerobic bacteria, which you would find in the respiratory tract. And anaerobic bacteria that you would see more deep in tissues like Clostridium. And then there are some that are facultative. These can spread more insidiously, especially if they're pathogens, because they can survive with or without oxygen. Okay. So then at the species level, you can have different forms of bacteria, different strains. The bacteria can accept DNA from unrelated forms via the pilus. They can alter their genetic makeup by other mechanisms. DNA in the chromosome is very fluid. It can excise out, can recombine. And so you have species variability actually within the species as well as intra-species variability. For example, E. coli is a species, but there are many, many different strains of E. coli, so there's a lot of interspecies variability. That's very important medically because there are certain strains of E. coli that are toxic and others that are not. So when we talk about subspecies, this is below the species classification. Sometimes they're referred to as strain or type. In terms of those bacteria that are bad and are obligate parasites, that means that they cannot survive without a host. Most bacteria do survive on their own without a host. The bacteria that we use in the lab, we can grow on artificial media, and it does not need to invade a host cell. However, a parasite, an obligate parasite, needs a host. Rickettsia, this causes diseases like Rocky Mountain spotted fever, rickets, typhus. These transmit via a insect host, like fleas, lice, and ticks, and they're transmitted to mammals. Rickettsia likes mammals better. This is where they can infect, whereas the arthropod or insect hosts are more of a vector, and they allow transmission. Insects don't get Rocky Mountain spotted fever. They don't get sick and die when they're infected. They just fly around and infect mammals. Chlamydias, this is a sexually transmitted disease, and it survives in the mammalian host and is transmitted in that way. Okay, here's Rickettsias. In endothelial cells, you can see some cell damage where it's staining dark blue. Here's chlamydia. You can see the individual bacteria in mammalian cells. Okay. Other things that bacteria can contain are thylakoids. You may remember thylakoids as a part of the chloroplast, but in the case of bacteria, they're prokaryotic, so they don't have chloroplasts. Chloroplasts are organelles. But instead, cyanobacteria, which are blue-green, contain these small compartments called thylakoids. The thylakoid membranes have chlorophyll, and they also contain gas inclusions, so they float to the surface and increase the light exposure. There's green and purple sulfur bacteria. This is a type of photosynthesis that does not give off oxygen. It is, by definition, photosynthesis because it's producing a sugar. And this is an interesting set of bacteria because they utilize sulfur. Not only do they photosynthesize to make sugar and not oxygen, but they also use sulfur in their metabolism. So you can see little yellow dots where there are actually grains of sulfur within the bacteria. Okay. Archaea. This is the third domain. Archaea differ in ribosomal RNA sequences. That's how they were initially identified as being very unique for bacteria. Their membrane lipids are different, and their cell wall construction looks more like eukaryotic cells. And we find them in extreme environments. Many of them are extreme, some of them are not. And those that live in extreme environments are called extremophiles. Methanogens grow in oxygen-deprived conditions. They breathe carbon dioxide and hydrogen gas, and they make methane. Halophiles will tolerate high salt. Psychrophiles will tolerate low temperatures, and thermophiles will tolerate high temperatures. So these are different forms of archaea. Here's some growth at Yellowstone. This shows an extremophile, a thermophile. This is a hot spring. So it's growing under high heat conditions. Okay, so let's review. The spherical bacteria is called, a rod-shaped bacteria is called, what's a phenotype? And name 4 types of medically important bacteria. What's a thylakoid? Okay, so spherical bacteria, hopefully you wrote these down. And now you can check your work. First answer is coccus. Rod-shaped bacteria, bacillus. Phenotype is an outward appearance of an organism. And there are many types of medically important bacteria. Gram-positive and gram-negative are medically important. Aerobic and anaerobic are medically important. Facultative are also medically important. Okay. And a thylakoid is a single membrane structure in cyanobacteria that contains chlorophyll and that allows the bacteria to photosynthesize. Okay, and then that concludes Chapter 4, and we will move on to Chapter 5. Chapter 5 deals with eukaryotic structure. And remember, this is in the context of microorganisms. So the different eukaryotes that we're going to look at include fungus, they include protozoa, algae, and helminths. So we're not looking at bacteria or archaea, we're looking at the 4 other classifications of organisms, and then which viruses, which are not prokaryotic or eukaryotic, we'll look at in Chapter 6. So what we're going to do is look at anatomy and physiology, look at the different families of these organisms, and understand the life cycle and follow the transmission of eukaryotic-based microbial diseases. Okay, so eukaryotic cell, organelles, and membrane-bound nucleus. There is evolutionary evidence that suggests eukaryotic cells evolved from prokaryotic cells via intracellular symbiosis. The hypothesis or theory is that prokaryotic cells were trapped in other larger prokaryotic cells, and then the smaller ones became organelles. The smaller ones that were aerobic bacteria became mitochondria. The smaller ones that were cyanobacteria became chloroplasts. First eukaryotic cells were probably single-celled. Single cells grow in colonies, but these colonies then presumably became specialized. And then multicellular organisms evolved, and instead of growing in a colony, they would grow in an organism structure where the individual cells would not survive alone. Okay, so here's an animal cell. This is an interesting, I don't like this cell. It's actually a plant cell, but it's very irregular. Plant cells don't look like this. They're usually much more regular in shape, squares or cylinders. This looks like an animal cell, but it has chloroplasts, so it's not an animal cell. We'll talk about all these organelles individually. Do you see a membrane-bound nucleus? And definitely organelles. This particular cell has a cell wall. You do not see cell walls in animal cells. We are not rigid. We need flexibility. So we'll start with appendages. Eukaryotic cells have flagella. However, they're different. They're larger than prokaryotes. They're structurally more complex. They're made out of microtubules. You may remember microtubules as the one of the structures in the cytoskeleton for eukaryotic cells. And flagella are comprised of 9 microtubules, bundles surrounded by 2 microtubules. And instead of rotating like prokaryotic flagella, eukaryotic flagella whip. So write that down. Prokaryotic flagella move like a propeller. Eukaryotic flagella whip. Okay. Eukaryotes also have cilia. And this is, you only find this on paramecium and then on some animal cells used for motility. The cilia will move in a coordinated fashion, and that allows locomotion. Okay, and here are some examples of some eukaryotic cells. These are euglena, and you can see the different types of flagella. We have all of these are polar arrangement, and some are instead of monotrichous, they're lophotrichous. Okay. Here is a human cell with flagella. We all know what that is. Okay. And here is a cross-section of cilia. And so you see 9 microtubule doublets. You can see the small circles in the individual cilia. 9 microtubule doublets that make a circle, and then 2 small microtubules in the middle of the circle. And you see this arrangement consistently in both cilia and flagella. Okay, these are cilia that are in lung tissue. The cilia and lungs are important because they will push foreign particles out of the lungs. Okay. There is a glycocalyx in eukaryotic cells. You can have a slime layer or a capsule just like prokaryotes. This is usually comprised of polysaccharides. This gives a biofilm, and so biofilms give protection. They allow the organisms to adhere to surfaces, and some of the sugars are involved in terms of cell signaling and cell recognition. Eukaryotic cells can have a cell wall, not all of them do. And all cells have a cell membrane. Algal and fungal cells have cell walls. These are made, for fungal cells, they're made out of chitin, which is similar to the molecule that you find in crab shells, and cellulose. Okay. Algal walls are highly varied in composition. We call them walls because of their rigidity. Okay. And then there are membranes. And the cell membrane consists of a phospholipid bilayer, just like you find in kind of ubiquitous in all organisms. Embedded are proteins and also sterols. Sterols, like cholesterol, provide stability. They provide flexibility when the cells get cold, and they also provide rigidity when the cells get too warm and become too fluid. And cell membranes are there for barrier function. They keep bad things out. They only let very, very few molecules in. Okay. Here's a fungal cell wall. You can see that the base layer is chitin, and then there are different types of molecules, sugars called beta-glucans, and then nanoproteins that are part sugar and part protein. Okay. Here's a component of a cell wall. This is for algae. This is just one example, alginic acid. Okay, so then we move on to internal structures. First of all, the nucleus. The nucleus houses chromosomal DNA. Okay. Endoplasmic reticulum transports material from the nucleus to the cytoplasm and out of the cell. Golgi apparatus is downstream of the endoplasmic reticulum. It also, with the endoplasmic reticulum, they both modify proteins through the addition of polysaccharides and lipids. Mitochondria are the cell energy powerhouses or power generators. Chloroplasts are the site of photosynthesis. And ribosomes. This is an odd thing to put a ribosome here because ribosome is not an organelle. Okay, so mark that down. The rest of these are organelles. A ribosome is a molecule. It's a molecule that takes messenger RNA and translates it into protein. And then there is a cytoskeleton. The cytoskeleton has 3 different components to it, and it supports structure. At the center of the cytoskeleton is a centriole. And there are 2 centrioles, and the 2 centrioles play a very vital role in terms of the microtubule structure and organelle structure. Here's a picture of the nucleus. The nucleus has an envelope. It's a double membrane envelope. There are nuclear pores that allow mRNA out and proteins in. And then there's a nucleolus. The nucleolus is darker, and there is chromatin in the nucleolus. That's the site of ribosome production. And then there are individual chromosomes. So the nucleolus, like I said, site of ribosomal RNA synthesis and collection of proteins for ribosomal subunits. The chromatin is usually dark fibers, and when we refer to it as chromatin, it's more of a mixed bag. It's not very, very tight chromosomes in mitosis. This is much more loosely arranged and much more frequently transcribed. Chromosomes are more tightly wound. You can see the individual chromosomes, the way that they're drawn, it looks like we're in phases of mitosis. Okay. And nuclear pores then allow for mRNA to go out, proteins to go out and in, ribosomes to go out after they're made in the nucleolus. And here's a quickie review of mitosis. I just want you to look at this. You've seen this before. You should know the different phases of mitosis. And that's it. Okay, not going to do the film. Here's the endoplasmic reticulum. The endoplasmic reticulum is attached directly as a large group of membranes is attached directly to the nuclear envelope. The endoplasmic reticulum is studded with ribosomes, and those ribosomes translate mRNA to make protein. The proteins then are secreted into the cisternal space. The rough ER is studded with ribosomes. The smooth ER is not. The smooth ER is more of a chemical factory for non-protein chemicals. Okay, and here's a small figure. This shows the ribosomes sitting directly on the endoplasmic reticulum. Several ribosomes are translating the same segment of messenger RNA. When the protein's synthesized, it goes into the cistern of the endoplasmic reticulum. Downstream of the endoplasmic reticulum is the Golgi apparatus. This is where proteins are modified and decorated with sugars. Some of the sugars are trimmed back, and then they're packaged to go into their final destination. Transitional vesicles are these small vesicles that are membrane bound that carry proteins from the endoplasmic reticulum to the cis face of the Golgi apparatus. Condensing vesicles then contain proteins and some of them contain lipids. And these are just little taxi cabs. They will transport the proteins to different organelles in the cell. They'll also transport proteins to the cell membrane so the proteins can be secreted. So if you look at these three organelles, the nucleus, the endoplasmic reticulum, and the Golgi apparatus, this comprises the endomembrane system. Okay? And the endomembrane system is responsible for transcription and translation of proteins. So in the nucleus, DNA is converted to messenger RNA. Messenger RNA is converted to proteins in the rough endoplasmic reticulum by the ribosomes. Proteins are stored and modified in the rough endoplasmic reticulum. Transitional vesicles then carry the proteins to the Golgi apparatus. Proteins are further processed where sugars are added and modified in the Golgi. And then after the Golgi apparatus, proteins are transported throughout the cell using condensing vesicles. If they're secreted, we call the vesicle a secretory vesicle. Other vesicles include lysosomes. Lysosomes are actually packaged in the Golgi bodies, and they specifically have enzymes that will digest large food particles. This is the stomach of the cell, and there are many lysosomes that will merge with vesicles that contain large particles of food. The vacuole, which you would find in plant cells, some protozoa, and I believe some other cells, some other eukaryotic cells, usually contains fluids and solid particles that have to be digested, excreted, or stored. And most often these substances have to be sequestered or put into a specific spot in the cell because they're toxic to the rest of the cell. In plant cells, the majority of the cell is taken up by what's called the central vacuole. So what you see here when you look at a plant cell is primarily the vacuole, and then chloroplasts are pushed up against the plant cell wall, as well as the nucleus. Over here is a contractile vacuole. Contractile vacuoles will swell, like you see in this particular photo, and when they swell, they then will move to the plasma membrane and release water from the cell. So contractile vacuoles help the cell get rid of water. Okay. Here's mitochondria. The bulk of the energy supplied to the eukaryotic cell comes from the mitochondria. And you'll see that we have the outer membrane and the inner membrane, and within the inner membrane are these projections called cristae. And cristae are the folds that contain the electron transport chain, which is responsible for cellular respiration. The inside, the jelly kind of liquid portion of the inside of the mitochondria is referred to as the matrix. The matrix contains mitochondrial DNA, and it also has ribosomes that look like prokaryotic ribosomes that allow the mitochondrial DNA to be translated into protein. And mitochondria, like chloroplasts, are almost like a cell within a cell. They have very few genes in their genome, but those genes are transcribed and translated directly in the matrix, and they also divide independently of the cell. So you get multiple mitochondria and mitochondrial divisions within a cell that's not even dividing. Here's a chloroplast. Chloroplasts are found in aquatic plants and algae. They convert sunlight into chemical energy.
Representatives are still helping other customers. Please continue.
photosynthetic role of chloroplasts helps to make organic nutrients not only for them, but for others that consume organic nutrients that aren't photosynthetic. The inner membrane consists of thylakoids, and these thylakoids are these small green discs. A stack of a disc is called a granum. Multiple stacks are called grana. And the thylakoid membrane contains chlorophyll. The chlorophyll pigment absorbs and transforms solar energy into chemical energy, and the chemical energy is converted into glucose directly in the stroma of the chloroplast. Stroma of the chloroplast is it's just the fluid that's encased inside the membrane. Chloroplasts also contain DNA and ribosomes, so they are able to divide independently, transcribe, and translate protein. Now let's talk about eukaryotic ribosomes. We talked a little bit about prokaryotic ribosomes and looked at their structure. There's a small subunit and there's a large subunit. Eukaryotic ribosomes function the same, and you know that that is the protein production factory of the cell. The large subunit is 60S. If you'll remember, the large subunit for prokaryotes was 50S, and the small subunit is 40S. S refers to Svedberg units, and the smaller a Svedberg unit is, the smaller the particle is. Okay. Total size is 80 Svedberg units, and it doesn't add up to 100 because it's based not on the actual molecular size, but it's based on the sedimentation rate. Okay. Multiple ribosomes will act on the same piece of RNA and translate it into multiple proteins. And when you have multiple ribosomes in a chain on a single strand of RNA, they're called polysomes. Okay. And here is a ribosome. The sort of darker gray, blue-gray up here is the large subunit. The lighter gray is the small subunit. Here is the messenger RNA, the 5' and the 3' end, the protein that's being produced, and transfer RNA molecules in the aminoacyl binding site, the peptidyl binding site, and the exit site. Okay? And here's a picture of some ribosomes that are studying an endoplasmic reticulum, and the endoplasmic reticulum is dyed green, the ribosomes are brown, and the cytoplasm of the cell is blue. Okay. There's also a cytoskeleton in eukaryotic cells. And this is a framework of molecules, primarily proteins, used to anchor organelles, move RNA in vesicles, and also involved in giving the cell shape and allowing for shape change. Microfilaments are the smallest filaments. They are made out of actin, okay, and they form a cell membrane, attached to the cell membrane, and are throughout the cytoplasm. And then there are microtubules. These are long, hollow tubes, and they help maintain the shape of the cell. Microtubules are also involved in the architecture of cilia and flagella, and they play a very strong role in mitosis in terms of making the mitotic spindle and allowing the cell's chromosomes to divide to make two daughter cells. Okay, so let's review. So, eukaryotic cell, in contrast to a prokaryotic cell, just two simple things. The eukaryotic cell has an enclosed nucleus, and eukaryotic cells have organelles. They are structurally more complex. the difference between flagella and cilia. Basically, flagella are longer and cilia are shorter and much more numerous on the surface of cells. Each one is made out of microtubules. Flagella move in a whip-like fashion for eukaryotes only, and cilia move in a coordinated oar-like fashion. Name the organelles of the cell. I'll just let you do that on your own. Name the organelles of the cell that contain DNA. Well, obviously the nucleus contains DNA. The chloroplast and the mitochondria also contain DNA. What do mitochondria do? They produce ATP. They produce chemical energy breaking down glucose. What do chloroplasts do? Chloroplasts do, they will uptake sunlight energy and they convert it into chemical energy by making glucose. Okay.
Our representatives are still helping other customers.
By the way, I'm on hold. If you wondered what the music is in the background, that's hold music. So I'm trying to multitask right now while I do your lecture. So some eukaryotic microorganisms. We're going to start out with fungi. This is my CDA. And includes two broad classes, molds and yeast. The majority of fungi are unicellular, so they exist as single cells or as colonial. Okay, these are colonial, grow in colonies like a colony of bacteria, so they're identical cells, they're just growing in a enclosed space. And there's two basic types of cells, yeast and molds. Okay, yeast are round to oval shaped cells and they reproduce by budding. Molds include hypha. Hypha is another term for mold. These are long thread-like cells found in filamentous fungi. You do have yeast that look like hypha, and these are called pseudohypha. They're just long chains of yeast cells. And then you have fungus that can go back and forth. They can take on the shape of either yeast or molds. Alrighty, and here's just a garden variety infective yeast. This is called Candida albicans. And this is sort of a skin fold yeast infection right here where you have some redness. And this redness, or it's also called erythema, is associated with swelling and tissue irritation due to an immune response because of the presence of the yeast infection.
Representatives are still helping other customers.
Here's a type of mold. You can see this is filamentous fungi. Okay, they make these long filaments. And this is a type of foot mold, athlete's foot. Okay, this is a tinea type mold. Tinea is the genus. And it's also infective. Now, in terms of fungal nutrition, fungus will eat just about anything. They're heterotrophs. They don't make their own food. They're not photosynthetic, so therefore they can't make their own glucose. And a heterotroph, as you know, is an organism that uses organic substances that it gets from others in order to get chemical energy. Many fungi are saprobes. They will feed on dead and decaying materials. Some are parasites, like a yeast infection or athlete's foot, and they exist on bodies of living hosts. And fungi are found in nutritionally poor environments. They really eat just about anything. So you're very, very easily can grow mold if you just leave something in the refrigerator too long. Mold, yeast and molds will grow in low temperatures, especially molds. And when you have a fungal infection, it's called a mycosis. And there are fungal toxins, some associated with peanuts, some associated with other types of molds, and they will produce spores, and these spores will cause allergies. Yeasts look a lot like bacteria. The colonies are soft and uniform. Molds are more cottony or hairy. And when you have hypha in a colony, molds in a colony, you call it mycelium. That's sort of just the term of mold growing.
Still helping other customers. Please continue to hold.
Separate hypha cells will form. Hello?